Hello friends, colleagues and viewers all over the world. You guys are following me on MuniMeterHealth. This is your source of learning cardiology and of course about human heart awareness and illnesses. I'm Dr. Jaitly. Welcome again on MuniMeterHealth, your source of learning cardiology. Remember, we are a medical education and patient education site where we strongly feel that the clinical outcomes will be enhanced if the two are combined together. This is not a medical advice channel, just make a note of that. And of course, if you need to seek any advice, must consult your own doctor. And if it's an emergency, get to the nearest hospital, please. And to my dear students, residents, and fellows, I always say that this is not a substitute to your syllabus. It may be a supplement to your syllabus. Please make a note of that also. So having said that, let's get into these short segments that we normally bring up for you. And today we are going to be talking about stem cells. Can they mend the broken hearts? Well, the broken hearts are broken hearts, but the fact is that we can mend them still. Let's see what we can do about them. Let's understand what stem cells are. Stem cells are the undifferentiated cells which have an inherent capacity, a very extraordinary, remarkable potential to differentiate into the actual organ cells, if you will, of an adult. Like they could differentiate into liver cells, hepatocytes. They can differentiate into cardiac cells, and therefore they could be myocytes, cardiomyocytes, and what have you, and blood cells like red cells and white cells and what have you. So these stem cells, they were present at the time when the fetus is being formed as an embryo, obviously, and then later on, even as an adult, they persist in pretty much throughout the body. So stem cells, so having known the uniqueness about the stem cells, let's understand how we can apply it to cardiology. Very importantly, well, first of all, we have to understand there are PCIs that are happening, there are CABGs that are happening, we have the thrombolysis that's going on, and we have the best conventional therapy right now to save the heart attacks, for instance, right? By providing the ischemic area with the actual blood supply through these routes, either by a balloon angioplasty, a stent, or by grafting, by doing the CABGs, or even by thrombolysis, where you can actually lyse or dissolve the clot out from the infarct artery. So having understood these approaches, we still have to we still are looking for a novel approach where we can actually replace a cardiac myocyte by a cardiac myocyte. So here's an example. This is the commonest analogy I give. These are the various bricks. Now, would you rather have something, if there was a problem here, would you rather have that replaced by something softer than a brick or something which is not as functional like a brick? Well, the point I'm making is cardiomyocytes, these are cardiomyocytes. This is where the infarction has occurred, and as a result, this is replaced by a scar tissue. Now, when it's replaced by a scar tissue, this is non-contractile. It is a non-contractile. In other words, here is your left ventricle, okay, LA, and this is your LV. And if this was the area, for instance, under jeopardy, which suffered a heart attack, then at that point, you could have done and brought a new graft, which will be the CABG, or you could have actually opened up the artery by putting a stent. If you opened up the artery by putting a stent, you can still restore the blood supply and hopefully save the portion of the heart attack that's from occurring any further, correct? But you will not be able to replace cell by cell the damage that has already occurred. So along came the novel approach, and again, this stem cells have been around for ever since medicine has evolved. I would imagine we knew about the stem cell because they are the original embryonic cells. But the fact is that the stem cells have found their way in oncology for almost 20 years, and they have been clinically applied to unfortunate pediatric age groups, specifically so for bone marrow transplantation in the leukemics, etc. Again, coming back to cardiology, that's where the interest has begun. So the resurgence of stem cells into cardiology has only happened in the past 8 to 10 years, where numerous studies have happened specifically for ischemic cardiomyopathies, for dilated cardiomyopathies, and post-MI patients, where they have started to think that perhaps the heart failure that's resulting from these entities, these clinical entities, can actually be benefited by applying stem cells. Well, before we apply the stem cell, let's find out how do we go and get them. So stem cells will have to be found in 3 sites. There are 3 sites. One is obviously the embryonic, which is not the best way to really, but amniotic fluid can still provide good stem cell substrate. Mesenchymal cells, or the one that's originating from the bone marrow, for instance. Bone marrow and blood is extremely rich in stem cells. So bone marrow is easily obtainable because you can go from the iliac crest or from the femur perhaps and get the bone marrow aspirations done and extract stem cells. Likewise, from the blood, you can do a blood, it's called apheresis, and the stem cells are separated and the rest of the blood is re-injected back into the donor. Okay, so these stem cells are autologous. Remember that they're coming from the same body, from the same human body, from the same person actually. So they have all the genetic background exactly the same. So there is no risk at all. And the third variety is the resident. We call it a resident stem cell, and these resident stem cells, they actually are present right here, RSC. They are the local resident stem cells which are present at the time of the infarct. So they were already there and they try to regenerate, just like there was a hepatocyte or a liver a portion of the liver that got damaged. How will that be regenerating? It will be generating via the stem cells of the hepatocytes. So those stem cells unique to hepatocytes will generate that portion of the liver. Same thing is happening with the heart as well, but not to the extent. And because it's not happening to that extent where it can replace brick by brick, in other words, brick for brick, or cell for a cell, in other words, basically replacing exactly the number of cells in order to restore the ejection fraction, came the novel approach that we should apply the stem cells to these areas. Now, how do you deliver? So once we find out what the source is, how do you deliver them? Well, the way you can deliver them is by either intravenous route. So here is your inferior vena cava, so they can go via the IVC and you could deliver it into the right atrium, etc. Okay, so that is one way to deliver it. The second way, so that's an intravenous route. So we'll write it as IV. The second route is the intracoronary route, IC, where obviously you have the coronaries, and we'll draw the coronaries here for a second. This is your heart, and of course the coronaries can be shown in the red. That'll be nice and good. Let's do that. And here's your left anterior descending artery, and from the left main also this becomes the circumflex. And there are numerous branches coming out here, as you know very well. Not a great sketcher here. This is your right coronary artery. All right. And the intracoronary can be also found. And the third variety is the intramyocardial actually. My pen is now showing some signs of sluggishness here. Anyway, so intramyocardial. So intravenous, intracoronary, and intramyocardial. So either of these three can actually be the routes. And the beauty is that they can all be applied clinically to these ischemic cardiomyopathics, dilated cardiomyopathics, and the post-MI situations where these patients can actually benefit from. So just so that you know, the heart failure that results where the ejection fraction, say, was 50% to begin with, has now dropped 50%, has now dropped, say, now to 45% or 40%, and as a result, you do have a drop in the ejection fraction in these patients, and that's because the portion of the myocardial infarction that has caused a regional dysfunction, if you will, or absence of function or dyskinesia in that area, and as a result this scar tissue that has developed has contributed to the dropping of the EF. So the whole idea is wanting to see if you can restore the ejection fraction back to 50% or to normal, whatever that number arbitrarily was, so that the heart failure progression is lessened or actually nullified or is nulled. So you can only do that by replacing cell for cell. That's what stem cells do. Okay, so this is a great novel approach. I thought I'll just discuss that very briefly in this short segment. And primarily, my goal is only to give you some basic information about various subjects that we bring up. And again, we like to stay